Welcome everyone. I hope you will enjoy this short video where I speak about biosolar cell and algae for a green economy. Over the last couple of decades, the environmental pollution has been one of the major problems affecting humanity. The pollution afflicting the Earth is leading to a severe climate change that might be soon irreversible. The pollution is mainly caused by an extensive use of the fossil fuels, which are still the basis of our economy and our lifestyle. However, this source of energy, besides causing serious pollution to the planet, will not last by the end of the century. This is not only an economic problem, in fact it directly affects the health of the world population, and continuing to use the fuels will only exacerbate the environmental and health conditions. One of the possible solutions is the transition to a green and circular economy model. The green economy is a theoretical model of economic development in which the growth is linked to an assessment of the impact that human actions have on the environment. Precisely, it is a form of economy that provides investment in favor of action that aim to achieve greater energy efficiency, reduce carbon dioxide emission, and protect the ecosystem. To this end, the use of renewable energy sources and the explosion of energy efficiency is of fundamental importance. In fact, the main challenge for the present civilization is to design innovative technology that are capable of addressing the main social, economic, and environmental challenges, such as globally increasing energy consumption and the necessity to reduce the carbon footprint in the environment to mitigate the climate change. The production of high-efficiency biohybrid photoelectrodes inspired by nature for powering the planet is a highly promising approach for environmentally friendly energy production. The scope is to convert light energy to green chemicals or electricity. Indeed, it has been estimated that energy captured in 1 hour of sunlight is equivalent to annual global energy produced by human population. But is it possible to use the sunlight for electricity production? It is well known that plants, algae, and some bacteria evolved in order to use the limitless and free sunlight for power their cells. This process is called photosynthesis. The attractiveness of the biophotovoltaic and solar-to-chemicals device resides in the potentiality of the photosynthesis process, which was improved all over 2.5 billion years. Understanding how to rationally manipulate the early stages of the natural photosynthesis process to convert the sunlight to storable chemicals energy in form of fuels and chemicals carries a great potential for developing a green technologies for solar to chemical conversion with minimal or even negative carbon footprint. For this purpose, the highly efficient natural proteins involved in photosynthetic process, such as photosystem I and photosystem II, are exploited in the semi-artificial photosynthetic system. But let's make a step back and say a few words about the photosynthesis process. The photosynthesis is a process used by plants and other organisms to convert light energy into chemical energy that can later be released to fuel the organism's activities. This chemical energy is stored in carbohydrate molecules, such as sugar, which are synthesized from carbon dioxide and water. In most cases, oxygen is also released as a byproduct, generating the atmospheric oxygen. Since in oxygenic photosynthesis water is used as the electron donor, the equation for this process is the following. In plants and algae, photosynthesis takes place in organelles called chloroplasts. A typical plant cell contains about 10 to 100 of chloroplasts, mostly in the leaves, although the organism we investigate contains only one chloroplast. The chloroplast is enclosed by a membrane, which is composed of a phospholipid inner membrane, phospholipid outer membrane, and an intermembrane space. Enclosed by the membrane is an aqueous fluid, called stroma. Embedded within the stroma are stacks of thylakoids called grana, which are the site of photosynthesis. Embedded in thylakoid membrane are the integral and peripheral membrane protein complexes of the photosynthetic system. In photosynthesis, the light-dependent reaction takes place on the thylakoid membrane. The inside of thylakoid membrane is called lumen, and outside thylakoid membrane is stroma, where the light-independent reaction takes place. There are four major protein complexes in the thylakoid membrane: the photosystem II, cytochrome B6F complex, photosystem I, and the ATP synthase. These four complexes work together to ultimately create the products ATP, the energy cone of the system, and the NADPH, the reducing equivalent. Plant absorbs light primarily using the pigment chlorophyll. The green part of the light spectrum is not absorbed, but is reflected, which is the reason why most of plants and algae are green in color. Besides chlorophyll, plants also use pigments such as carotenes and xanthophylls. Algae also use chlorophyll, but various other pigments are present, such as phycocyanin, carotenes, and xanthophylls. These pigments are embedded in plant and algae in complexes called antenna proteins. For instance, in the studied organism, there is a Phycobilisom's umbrella-like antenna complex. In such proteins, the pigments are arranged to work together. Such a combination of proteins is also called light harvesting complex, which structure depends on the species and environmental conditions. In photosynthesis process, the light-dependent reaction begins in photosystem II, when a chlorophyll A molecule within the reaction center of PS2 absorbs a photon. An electron in this molecule attains an excited energy level. Because the state of the reaction center is very unstable, the electron is transferred from one to another molecule, creating a chain of redox reaction, called electron transport chain. The electron flow goes from photosystem II to cytochrome B6F to photosystem I. In photosystem I, the electron gets the energy from another photon absorbed by the reaction center, that is a heart of the photosystem I. The final electron acceptor is the NADP. In oxygenic photosynthesis, the first electron donor is water, creating oxygen as byproduct. Photosystem II is extremely complex, a highly organized transmembrane structure that contains a water-splitting complex, chlorophylls and carotenoid pigment, a reaction center P618, pheophytins, a pigment similar to chlorophyll, and two quinones. It uses the energy of sunlight to transfer electrons from water to a mobile electron carrier in the membrane called plastoquinone. Plastoquinone in turn transfers electrons to cytochrome B6F, which feeds them into photosystem I. The step where electron goes from the water to the reaction center P618 is performed by a poorly understood structure embedded within the photosystem II, called the water splitting complex or the oxygen evolving complex. It catalyzes a reaction that splits water into electrons, protons, and oxygen. The photosystem 1 complex is an excellent macromolecular nanomachine, able to absorb photons in its photochemical reaction center and convert them into long-lived charge separation state. The quantum efficiency of the primary charge separation in the photosystem 1 complex is 100%. The need to find new sources of energy production for our everyday consumption is one of the most pressing issues of our age. To overcome the dependence on fossil fuel and to access renewable energy sources, innovative paths such as artificial photosynthesis must be followed to go behind the conventional technologies already available. A different approach to this problem is to take advantage and mimic what nature was able to perfect upon billions of years of evolution. Yet, one of the biggest challenges in modern science is to replicate in an artificial way the most basic process performed by nature, such as photosynthesis. This fundamental process has been found to be extremely complicated and very difficult to be reproduced in laboratory settings using artificial man-made systems or devices. Biohybrid devices are the meeting point of two disciplines that only recently started to interact, namely biology and material science. The potential of such collaboration is to take advantage of the best of the two fields and design and build devices that mimic the photosynthetic apparatus of plants in a laboratory setting. To do so, at Solar Fuel Laboratory, Centrum of New Technology, University of Warsaw, we interfaces protein which can efficiently harvest light upon immobilization on graphene substrate. This project proposed to use the highly robust photosystem 1 complex from an extremophilic red alga, Synadoschiton merola, since it has been proved to possess remarkable robustness and high activity, a prerequisite for efficient and long-lasting photoconversion in the semi-artificial devices. The light absorbed by the photosynthetic protein, purified, is converted into electron flow toward electron, providing the basis for a variety of biotechnological applications, such as solar-to-fuel. In particular, in the project, we are focused on the minimization of the charge recombination at the interfaces between the working modules of the devices. This is critical to boost the performance of these highly promising solar converting devices. Switching to this model of economy does not only mean using renewable energy sources, but also being able to remedy the pollution already caused. Indeed, another essential challenge of this century is to design an efficient and biocompatible procedure to decontaminate the polluted groundwater and contaminated soil. This involves the study and development of an effective bioremediation system that would allow to detoxify contaminants in the soil and other environments. In particular, bioremediation is defined as the process whereby organic wastes can be biologically degraded under controlled conditions to an innocuous state, or to levels below the respective concentration limit, as set by the controlling authorities. In other words, bioremediation employs the living organism, most notably microorganisms, to degrade the pollutants and convert them into less toxic or non-toxic form. The suitable organisms can be bacteria, fungi, or plants, which have the physiological abilities to degrade, detoxify, or make the contaminants harmless. In some occasions, the organism can be already present on site, indigenous microorganism, or can be isolated from elsewhere and added to the treated material, using bioreactor as an example. The effectiveness of this method is highly dependent on the applied environmental parameters that influence the microbial growth and the degradation rate. Bioremediation technique depends on having the right microorganism in the right place under the suitable environmental conditions in order for degradation process to occur successfully. The biological process involved in bioremediation technology can be enhanced and focused on the removal of specific hazardous pollutants from soil, water, or atmosphere. It can mineralize waste material into water, carbon dioxide, biomass, or other non-hazardous products and thus obviate the need for further treatment. Bioremediation can treat a wide range of compounds. Apart from municipal waste and processed water, suitable for each case, microorganisms may be also applied for the degradation of pesticides, industrial chemicals, components of crude oil, and even compounds that, until recently, were regarded as non-biodegradable, such as chlorinated solvents, chlorofluorocarbons, and other synthetic organic compounds. To this end, in our laboratory, we are using Cimerola and Disulpharia, both extremely red algae from the Cyanidales family, for this purpose. The order of Cyanidales is composed of asexual unicellular red algae that are known to grow in low pH environments and at moderately high temperatures, up to 56 degrees, and are typically found in acidic geothermal habits through the earth as volcanic hot springs. No other photosynthetic microorganisms are known to inhabit this combination of conditions. The order of Cyanidales, since 1981, is thought to consist of three genera: Cyanidium, Galgieria, and Cyanidoschium. These microalgae, therefore, have the ability to live in very inhospitable environments in which are present heavy metal due to their strong metabolism. The study of the detoxification of these algae in the presence of heavy metal could lead to a breakthrough in the treatment of water polluted by these contaminants.